welcome Dr. Brandon to our Wallace Coulter seminar series. And that's, the knowledge that we receive funding for this seminar from that foundation. Dr. Brandon got a bachelor in the University of Connecticut, and in, I think it's neuroscience? Psychology, yes. And a PhD in Boston University in neuroscience. Had a postdoctoral term at UCSD, and have been for some time, I think from 2015, assistant and associate professor at McGill University. His expertise is understanding how memory is formed and how mammalian in general navigate and understand the space. And he's, giving us a talk about his main achievement in this year. So, join me in welcoming Dr. Brandon.
Yeah, thank you for that nice introduction, and thank you for the invitation to come down here. I should have come. I'm in Canada now, so we should have come in the winter. So I'm experiencing a little bit of shock when I'm outside these days. So the title of my talk is the neural mechanisms of spatial cognition, and really the talk is going to be divided into three subtopics. First, I'm going to talk about the laser doesn't really work.
Does not?
So first I'll talk about the head direction system, reporting population dynamics and what head direction cells do when animals reorient or sort of refine their way when they're lost. I'll talk about an optogenetic study where we've been looking at the medial septum. This is a basal forebrain structure that's projecting to the hippocampus. We've been studying this, particularly this GABAergic long-range projection that supports grid cell firing. I'm using a lot of terms, but I'm going to introduce a lot of these things at the beginning of the talk. And then finally, I'll talk about some of our work in Alzheimer's disease, where we've been looking at these spatially tuned neurons in a mouse model of Alzheimer's. So I start with this statement that navigation is hard. And maybe it's not so hard nowadays. We all have phones in our pockets, we have Uber, it's pretty easy to get around. But I'm old enough to remember when I would go to New York City for the first time, for example. You go from your hotel to a restaurant. You need to take the metro, so you study a map. You get on the metro, maybe you need one or two different metro lines. And when you get out of the metro, you're coming out from underground, you know that you need to go north from that metro stop to get to the restaurant, but you're completely disoriented. New York City knows this, and they started putting these compasses that give you north, south, east, west to let you reorient yourself. And we wanted to study this process of when you're lost or you don't know which way is north, which way is the cafeteria, how do I get home? How does the system, how do our brains handle this problem? There turns out to be a completely dedicated circuit in your hippocampus. The hippocampus is known for learning and memory, but also for navigation. And if you record from neurons in the hippocampus, you see neurons that are tuned for space. So in the early 70s, John O'Keefe discovered, I'm really trying to use this laser pointer, discovered, I can just walk up to it, hippocampal place cells. So this was published in 1971. It won the Nobel Prize in 2014. So in black is just the path that an animal is taking in a square box. And the red dots are the location at which a single neuron spikes. So you can see this one neuron in the hippocampus likes to fire in this southwest corner of the box. A little bit later on, people were recording outside of the hippocampus and they discovered head direction cells. So these neurons, these are firing everywhere in the environment, but only when the animal faces in a particular direction. And then in 2005, the Mosier group in Norway discovered grid cells, and I'll be talking about them toward the end of my talk. These are like place cells, except they fire in multiple locations, and the locations are organized by an equilateral triangle. So a lot of my research is trying to understand how these neurons function, what they do, and how they dysfunction in disease. So I'll start off with the head direction system. So trying to understand what those neurons are doing and how they help you reorient when you're lost. There's a huge circuit in the brain starting in the vestibular nuclei, sending vestibular signals up to the thalamus. Here's the anterior dorsal thalamus. And then this is projecting up to the cortex, where you can have grid cells and place cells. But it's really coming up from the vestibular nuclei. And the main center of the head direction system is this anterior dorsal nucleus of the thalamus. So we'll be studying this nucleus. To do this, we use what we call a miniscope. I'm sure you've all used microscopes. We've taken this microscope and shrunk it down so that it's only 2 grams. It can sit on the head of a mouse as he's running around, and you can look down through the microscope into the brain and look at neurons firing and their activities. So this is a video that just played of what the data looks like. This is all done by Dr. Zaki Ajabi, a recent graduate from my lab, and you can see this video of the neurons flickering. We have to express a calcium indicator in these neurons so you can see them, but it's relatively straightforward to do this. So this is the anterior dorsal nucleus of the thalamus. We have a lens coming down to sit just above it. We're expressing our calcium indicator here, and then this miniscope just sits on top of the animal. This allows us to record from large populations of neurons. So you could use electrodes, but you might only get 5, maybe 10 neurons simultaneously. With this approach, this is the head direction cells all color-coded for their preferred direction. We can get hundreds of head direction cells, so we can really understand what the system is doing in those moments of reorientation. Okay. So one analytical approach that we take is called decoding. So at all moments, we can tell where the brain, where the head direction system is focused. You can, the way that this works is you feed the model about, you need about 100 or 150 neurons. You feed the model and you teach the model that when these neurons fire, that the animal is facing in this direction. When these neurons fire, the animal is facing in this direction. And so you can, after you teach the model, you can then decode the actual position. So in blue is the measured head direction. So we have a camera mounted above the animal. We can see where the animal is facing. And then in red, we have the decoded. So just analytically, we can guess with great accuracy where the animal is facing. This is important because we want to guess where the animal is facing during these moments of reorientation to watch the process happen. Okay, so we want to look at reorientation, alignment of the head direction network, independent of the animal actually moving its head. So we conceptualize the head direction network as a ring attractor. So you can imagine you have a ring of neurons that are aligned like this. The neurons next to each other are connected, and they're all head direction cells. So in this case, the bright colors or the red colors indicate maximal firing rate. When the animal is facing this way, these neurons are all activated, and these neurons are all silent. When the animal turns its head, you get this propagation of activity, this rotation of activity around this ring attractor. And we wanted to ask, in moments of reorientation, where the animal is not actually moving, but mentally is rotating the world around him, do we see these rotations even though the animal is not moving? This is our first question. And then we have the second question, which I'll get into, is how does the system do this? Perhaps it needs to lower the overall energy in the system to allow the system to rotate through this ring track. Okay, so how do we do this? How do we create these reorientation events in a mouse? We have the mouse sitting here with this miniscope on, reporting from the anterior thalamus. This is a 360-degree television screen that's surrounding it. There's a dome on top, and we have this camera watching we just play this one cue card that sort of polarizes, gives him a sense of where he is, or a sense of direction in the environment. And because it's a TV, we can start now playing with this cue card. We can take it away, and when we start to get lost, bring it back, he can reorient to it. We can also play a trick where we show him this cue card for 5 minutes, we take it away for 2 minutes, and then we bring it back, but we actually shift it. So now we're really forcing the system to reorient, to potentially shift through all of these head direction states. And so this is what the experiment is. We do a 0, this is just the baseline, the cue is here, there's a period of darkness, and then we shift the cue 90 degrees. We do this over and over again, and I'll show you exactly what happens. What I'm plotting here is the offset. This is the difference between the measured head direction with the camera, look at the camera mounted above the animal, and the decoded position that we're getting from our decoder. So when the cue is available, the offset is 0. Everything is working perfectly. There's been no shift or rotation around this ring attractor. The key here is what happens when we now bring this cue up, but shift it by 90 degrees. What you see is a rotation. It's a pretty slow rotation. It takes roughly on average about 30 seconds. But you see this rotation of this activity exactly 90 degrees. We can keep doing this. So this is a rotation through head direction states. And every time we re-show this cue shifted 90 degrees, we get this rotation through these states. So what you have here, going back to this video, is we put the animal now in darkness. The system is relatively stable. We bring a visual cue, shifted 90 degrees. And even though the animal isn't moving, you can see this activity is rotating through these states. So how does it do this? And this is a big question that my lab is now after. But the first hint that we had, okay, so it rotates. The first idea that we had was perhaps in order to get from here to here, it's difficult. And the reason it's difficult is because you have, these neurons are all active, they're synapsing onto inhibitory neurons, and those inhibitory neurons are counteracting all of the other neurons. So there's a competition here. These guys are winning. How do we rotate activity through this space where you have this active competition? And our idea is that maybe the system would drop down to some intermediate states, lower the activity, lower that feed-forward inhibition, so that the system could then rotate through the states. That was the general idea, and it turned out to be right. The way we did this is now these are reorientation events. These are the shifts through space on a single trial level. What I showed you before was the average across all of our sessions. And you can see that sometimes we have very fast rotations. Again, this is offset on the y-axis. We see these very fast rotations, so the animal is very quickly reorienting. In other cases, we have much longer ones. Here's one that's 30 seconds, here's one that's 45 seconds. So we thought, okay, can we use this variability to test this idea? Perhaps when the system rotates really fast, it's dropping down to these intermediate states quickly. Perhaps when it's going very slow, it's not going down to those intermediate steps. So we separated our data into fast and slow resets. And we thought maybe these fast ones would be going down to a lower energy state. And so we measured something we called gain. It's really just the overall activation of the entire network. You just look at firing rate, you see ups and downs in the firing rate. That's what gain is. And we see this massive drop down to about 65% of the firing rate, all of the neurons, when the system rotates very fast. And for the slow rotations, we still see a drop, but it's just not as complete. And we think this is the reason why it's taking the system so long to rotate. So even if you just look at the rotation speed here, we call it drift speed, as a function of gain, there's a very nice relationship. So really, we think that this has to do with lateral inhibition or feed-forward inhibition. Where if you're in state A, and A is very active, lots of activity, you're recruiting quite a bit of this lateral inhibition that's preventing you to transition states from A to B. But if you just lower your activity of A, it's going to reduce the amount of lateral inhibition, which will make a transition between states much easier. So we can model this pretty easily. So this is what the data looks like in just a different format, where I have, it's a bump of activity that starts at 0 and is rotating because of the reorientation event, rotates to 90 degrees. These are the fast ones, these are the slow ones. And you can see for the fast ones, there's a bit more of a dip here in the overall bump of activity. Maybe less so here. But we can model this by just taking what we call gain modulation. It's just an inhibitory input to all of the head direction cells in the network to lower the overall activity. It allows the model to rotate fast when you supply it with high gain. So a lot of the work in my lab now, this is all published, but a lot of the work in my lab now is trying to understand the function of gain. Perhaps it's actually signaling something about the certainty of the representation and also the source of this gain. Is there an actual direct inhibitory projection into the ADN, into the thalamus, that's helping the system reorient when animals are lost? So just in summary, we have a reorientation involves a rotation around head direction states, reduction of network gain, this population activity metric. The more you reduce the gain, the faster the system can reorient, the more flexible it becomes. And we hypothesized that this is a result of just less lateral inhibition, less competition between neighboring states. Okay. I feel like I can take questions here before we move to the next. Yes?
So how stable, I don't know if you've studied this or anybody has studied this, but how stable is the cell's preference over time?
Yeah, great question. It's incredibly stable. In our hands, we never see any like representational drift. In place cells, you actually see, they're stable across a few days, but if you go out a week or two, they're just constantly changing. Head direction cells seem like they're stable forever.
Okay.
So, yeah, when this is rotating, is the Q rotating or is the flap?
So it's just the Q that's been rotated 90 degrees. So it would be like if you were convinced that the parking lot was over there, and you just had this idea in your, you think about the layout of this building, and you have a certain map, you have a certain sense of direction, but then you realize that you were wrong the whole time, and the parking lot is actually over there. You would take a moment to have to sort of rotate your entire map, and you sort of be confused for a second, and then you rotate your map, and that's what's happening with the mice, is that they're just rotating.
So rotating mentally, internally, but no, because I'm thinking when you're rotating your head, the vestibular system may be sending some signals to the hippocampus to tell you information about where you're.
Yeah, for sure. So this whole time,
Acceleration at least.
So this whole time the animal's free to move, and you have activity going around this ring attractor. But on top of that, when you reorient, that whole thing gets shifted by 90 degrees.
So you don't have any system in place like to keep like the rats that you're observing like in the same orientation like while you're moving the Q curve? Because I know you mentioned that like you could see like the directional change, even though like they didn't move.
Yes.
Like when the Q moves.
Yeah.
But like so you're not doing anything to keep the rat still though?
No, the rat's free to move at will. If we head fixed him, which is possible, you'd see the same amount.
Perhaps I was misunderstanding, but so that initial experiment you were showing where you set a Q at 0 degrees, turn the Q off, waited several minutes, then had 90, and then turned it off. So when you turned off at 0, like that it was stable at 0.
Yeah.
When you turn off at 90, actually like it was kind of declining very slowly back down.
Yeah, that's a nice observation. So we report this in the paper, and we call it this reversion, where you've tricked the animal into rotating that 90 degrees, but when you take the Q off, there's something in the system that reverts back to the original Q location. At first we thought that maybe it was continuing in the same direction, so if you rotate 90 degrees and then you turn the lights off, maybe it will keep rotating that way, but it's not, it's actually going back to the
So that's a different memory of that initial.
It's either a memory or, I mean, we can't control for all sensory stimuli. So maybe there's something, in the chamber, or there's auditory sounds coming from the hallway or something like that that might be influencing the system. Okay, so we'll jump to this second part of the talk, this long-range GABAergic projection from the medial septum. So let me first tell you a little bit about the medial septum and the hippocampus. So the medial septum is this basal forebrain structure, sits right on the midline. It's a very important structure for the hippocampus. So the hippocampus, I'm sure many of you know, is important for learning and memory. If you lesion the hippocampus, you can't make new memories. If you lesion the medial septum, you can't make new memories. Lesioning the medial septum mimics an entire lesion of the hippocampus. It sends a massive projection to the hippocampus, and it degenerates in Alzheimer's disease. There's lots of reasons to be studying the medial septum. When you're recording the hippocampus, you see this large oscillation called theta rhythm. It's actually the largest amplitude rhythm that you can record in the rodent brain. And I know Tim has been reporting lots of interesting things on theta oscillations. They're observed whenever the animal is navigating, solving memory tasks, otherwise just being attentive in the world. You see theta in the hippocampus. It reflects the rhythmic spiking of neurons. So if you look at theta oscillations and then you look at the spiking of neurons, it's because all of the neurons are very highly organized at this frequency. If you lesion the medial septum and you record from theta oscillations in the hippocampus, you completely wipe out this rhythm. So this is some work I did in graduate school. If you record in the hippocampus, you see very nice theta. If you inactivate the medial septum, in this case this was Musmol, it's a drug that acts on GABA A receptors, shuts down all activity in the medial septum. We shut down theta oscillations in the hippocampus. What we wanted to do, and this is an old study that I did as a graduate student in Boston, we wanted to ask, when you lesion the medial septum, what happens to all these spatial cell types that I told you guys about, the head direction cells, place cells, and grid cells. So we set out to do this. We put, it seems like archaic technology nowadays, but this is a cannula, metal cannula going down into the medial septum so that we can just inject Musmol directly into it, some recording drives and some electrode tracks in the entorhinal cortex. And we started off by looking at these grid cells in the entorhinal cortex. And what we found very quickly is that when you inactivate the medial septum, the firing of grid cells is completely disrupted. So here you see the firing fields of this particular cell. We hold on to it, we record it when the medial septum is inactivated, and the grid cell no longer fires like a grid cell. Not true for head direction cells. Head direction cells don't seem to care about inactivations of the medial septum. And also not true for place cells. Place cells in the hippocampus are totally intact when you infuse Musmol into the medial septum. So the medial septum seems to really be supporting theta oscillations and grid cells in the entorhinal cortex. So that was all well and good, and that was work that I did as a graduate student. But when I started my lab, we started using move to mice. We have transgenic mouse lines. We have optogenetics. We can do this study, but be a bit more specific with it. So the medial septum actually contains 3 types of neurons. There's GABAergic neurons, there's glutamatergic neurons, and there's cholinergic neurons. All 3 are projected to the hippocampus. And in our previous work, we inactivated all of them. Didn't differentiate, we just injected mu small, and all of them were inactivated. Here, and this is work led by Jennifer Robinson, we targeted just the GABAergic interneurons. So we used a VGAT Cree mouse. This is a mouse that Cree is expressed in all GABAergic neurons, and a Cree-dependent virus to drive expression of ARCH-T. So this means when we insert a fiber optic and put light into the medial septum, we can selectively silence just the GABAergic cells in the medial septum. So this is what the surgery looks like. This is our fiber down in the medial septum, and expression of this opsin ARCH-T. You can see when we turn the laser on to activate ARCH-T, and we're just shutting down the GABAergic medial septal cells, in the entorhinal cortex, we see this reduction in theta oscillations, so it seems like it's really working well. And even when you turn the laser off, the theta oscillations return, and they return immediately. So we just asked the question, what happens to grid cells? Is it the GABAergic projection from the medial septum to the entorhinal cortex that's responsible for supporting these grid cells? And the answer is yes. So when we turn the laser on, these cells still fire, but no longer in the beautiful hexagonal array. You can see some of these fields are even being turned off. So the gridness, this is just a measure of the spatial periodicity of the grid cells, is reduced. And what we even noticed is we would do this inhibition for 30 seconds, and we were looking at the evolution of grid cell firing. I told you that theta oscillations return right away. But the grid cell firing actually takes about a minute, a little over a minute to recover. So we're doing some sort of long-term damage to the dynamics in the network. So just a brief summary. So optogenetic inhibition of these GABAergic cells, it's reducing the power of theta oscillations, and it's disrupting the spatial firing patterns of grid cells. So continuing our focus on grid cells, one of the things that I wanted to do when I started my lab is not just ask fundamental questions about how circuits are working, but how does this relate to disease? So we started working with mouse models of Alzheimer's disease. In Alzheimer's, you have 2 main culprits. There's amyloid beta plaques, which begin to accumulate, and tangles. And in our lab, we started focusing on this first one, the amyloid beta plaques. So we chose this J20 mouse model of Alzheimer's disease. It's a very aggressive model. These animals start developing plaques, very small plaques, at about 6 or 7 months, and by 8 or 10 months, we start seeing widespread plaques throughout the brain. This image is actually a 14-month-old animal, and you can see plaques all through the entorhinal cortex. We didn't want to ask the question, what happens to the system after the plaques arrive? We know that there's going to be disruptions. We wanted to ask the question, what's the earliest disruption in the network? We know these animals are impaired in spatial memory tasks. What's the first thing that goes? What computation, you could say, is most sensitive in these Alzheimer's mice? So we set out to record all of these cell types in these mice. This is work done by Johnson Ying. It was an incredible amount of work. We started implanting the animals at 3 months of age, when there's no plaques, animals are cognitively fine, and up to 7 months, where you start to see the plaques build up. So we recorded from an ungodly number of animals, over 100 animals, to get thousands of neurons throughout the entorhinal cortex. And a separate data set we also recorded from the hippocampus. So it was a lot of surgeries by Johnson. So thank you, Johnson. So what does the data look like? So here, each row I'm showing a different animal group. So the top is non-transgenic young, the next one is non-transgenic aged, then APP young, and APP aged. And so these are the top grid cells that we recorded from each group of animals. You can see that the top row, all of those grid cells are beautiful. You start to see in this APP aged animal that these are included in our top grid cells. You can see these aren't as nice as the other ones. So we set out to quantify this as best we could. And so we used a grid score. We can talk about how that's computed, but it's just a measure of how nice the cells are following in this hexagonal grid pattern. And you can see that the APP aged animals, the grid scores are actually getting close to a completely shuffled distribution. So a simulated animal with 0 grid cells. So the grid score is shifting down, and if you just look at the age of the animals as a function of grid score, we see this negative slope. Not true for head direction cells. They're totally intact. Place cells are also completely intact. We then looked at the behavior of these animals, just to see if there's a spatial navigation deficit that corresponds to the deficit in grid cells, and that's exactly what we found. So this is a task where the animal needs to navigate in the dark, find some food, and return to his refuge. Animals in the healthy groups do this quite well, and the APP aged animals are not getting back to their refuge as easily. So just a summary of the findings: grid cells are really the first to be disrupted. Place cells and head direction cells seem to be fine. I think there's an interesting clinical perspective here for using fMRI imaging. You can actually detect something like the grid cell signal that people can now be using in Alzheimer's patients. So just a summary, we talked about population dynamics of head direction cells during reorientation. This long-range GABAergic projection from the medial septum to the entorhinal cortex is important for grid cell firing. Grid cells are the first spatially selective cell to show a disruption in mouse model of Alzheimer's. And if that wasn't enough material for you, you can follow all of my lab's work on YouTube, where we've been making some efforts in knowledge translation to explain sort of in more simple terms the papers that are coming out of the lab. Okay, and yeah, so I just want to thank the people involved. So my student Zaki Ajabi was instrumental in getting the miniscope up and running and working in the lab. He published the paper on head direction cells. Johnson Ying for the tremendous number of surgeries that he did for the Alzheimer's project. Jennifer Robinson, a postdoc in the lab, for her work on the medial septum project. To collaborators, the rest of the lab, and of course to funding. And thank you for your attention, and I'm happy to take questions.
Thank you very much. So, open the room for questions.
First question is, why do you think you don't get degeneration of the place and head direction cells that you're getting? They're getting affected by the amyloid plaques, right?
Yeah, there's no significant change to them. There's a slight increase in the firing rates, but the tuning of head direction cells and place cells seem okay. Other people have recorded much later in the disease progression, and they see deficits in at least the
base cells.
Eventually, they get disrupted. Eventually, everything goes though.
Yeah, but this maybe makes sense because a head direction cell is sort of a more direct input. It's kind of like if they're facing a direction, then the cell's going to fire. place cells maybe like that as well. Grid cells are like a coordination of activity that then leads to this firing pattern.
Yeah, yeah,
So it's more sensitive to disruption earlier on, is it?
yeah, exactly.
Well, how would that relate behaviorally? Like, I mean, what would you expect to see if you're looking at a deficit? So, you know, fMRI in a diagnostic, like, that would be great to use the tool, but they don't use it. Is there any behavioral measure that would indicate, like in humans, do they?
Yeah, so it's a very good question, and the reason we chose this task, this path integration task, is because we think that grid cells are needed to solve this task. And so people, even with subjective memory decline, these are people that go to their doctor and they say, I have a memory problem, I'm worried I'm getting Alzheimer's disease. They'll then give them a battery of memory tests, and they pass every one of them. And the doctor says, sorry, you're fine, stop worrying, go home. But very often these people, 2 or 3 years later, have mild cognitive impairment and go on to develop Alzheimer's disease. When people now test these people on path integration tasks, not memory tasks, they see deficits in path integration. So there's been a lot of work to, even with VR goggles, so that you can do this in a clinic, do some kind of path integration task with the doctor. And I think this is the way of going forward, in addition to maybe some fMRI type work.
Yeah, that's good, great.
You said the disruption's mostly to grid cells, right? The distribution of the plaques in your mice, are they particular to entorhinal cortex, like the progression of AD?
So in this mouse, they seem to start in the hippocampus, and mostly in dentate gyrus, and then you start seeing them in the cortex, but it's really not specific once it gets to cortex.
So does that mean something about the sensitivity of the grid network to disruption versus head direction and place?
Yeah. I mean, you would, if it just followed the plaques, then you would think, okay, maybe place cells should go first. But all of our recordings are before you even start to see plaques. So there's, soluble amyloid in the system that's, killing synapses, general toxicity to the whole brain. Place cells are fine, head direction cells are fine, but it's the grid cell computation that just seems the most sensitive. I'll also say that, when you do manipulations like the medial septum work, it's the grid cells that are sensitive. Grid cells are very sensitive to changes in circuitry in the brain. Place cells, not so much. You can lesion massive inputs to place cells. You can lesion most of entorhinal cortex, and you still have place cells. So it's just a very robust signal, whereas the grids are just more sensitive in general.
Question. I have one question that, if I think more philosophical question, these animals are nocturnal.
Yeah.
So the way that they could see when they have a marker of their head, in my opinion, may be on two ways. One is they use their vestibular system to estimate acceleration from initial point. So every time they move and they rotate, that makes a prediction.
Yeah.
Or if they have in place a regression model from the place cells to estimate what the orientation.
Yeah, both can be done.
So which one do you think is the most likely? I mean, have you
Yeah.
already tried to make a regression from existing place cells to predict the direction of the head?
So you can do that. We can decode animals' head direction just from place cells. Yes.
So
easily.
that's interesting.
So that information is there. If you lesion the hippocampus, the head direction cells are fine. So I don't think it's not like the head direction cells are depending on that input, but there's definitely some kind of collaboration between these systems.
Any other questions?
Quick question about your second part. What was the priori hypothesis of targeting GABA neurons? Because they should inhibit something, and why did you expect that if you silence something that inhibits something will inhibit the grid cells, or there is known something about interneurons, which I'm not just familiar with the literature.
Yeah,
What does that mean?
good question. So this projection is the largest projection coming out of the medial septum. It also, it's been known for quite some time that this is the projection that's mediating theta oscillations. So previous work, this is some work that I did as a postdoc, is if you optogenetically stimulate the GABAergic cells, you can drive whatever frequency you want of theta. So you could, theta is usually about 7 or 9 hertz. You could, at 16 hertz, you could optogenetically drive the GABA cells and you get 16 hertz theta in the hippocampus. So we knew that they were important for theta, and our previous work, pretty small, we really focused on this idea that theta oscillations were somehow important for grid cell function. And there's a lot of models that use these oscillations to create that grid cell spiking pattern. So for that reason, we knew that we could alter theta oscillations, and that would likely have an effect on grid cells, at least so we thought. We've now gone and targeted the other cell populations, the cholinergic and the glutamatergic populations in the medial septum. And it turns out that they have maybe subtle effects on grid cells, but really grid cells are intact.
Sorry, so you measured the offset of that ring attractor when you changed that visual cue. Was there any interest in doing the opposite, so changing the orientation of the body of the animal, but without any visual cues of a change in orientation?
So no visual cues and you rotate.
Like, you know, if you're blindfolded and you turn, your body somehow knows that you did make a turn and now you're facing a different direction.
Yeah, so the system will track that. So you'll get, so the animal's head turns just like that video I showed where the animal's turning its head and you see this activity propagate.
Yeah.
That will happen even in the dark.
Okay.
It's the cue coming on at a shifted angle is the thing that's sort of tricking the animal and causing the system to rotate.
Oh, okay, so you were trying to measure the confusion.
Exactly.
Does that happen with olfactory cues or auditory cues as well?
Yeah, good question. So olfaction would be difficult to control carefully. Auditory, I think, is an interesting direction to go. We haven't done it. I think we were so happy that the system is so driven by visual stimuli. Like, I didn't show you this, but one thing we do is we actually, instead of doing a shifted cue, we continuously rotate the cue around the animal. And the system will continuously be reorienting to that cue, so you can drive activity to go around this ring attractor. You can then turn the light off after we did this for 7 minutes, and then turned it off, and you actually see the system continues to rotate in that direction. The animal's totally confused by that. So visual stimuli seem so strong at manipulating activity in the network. But my students keep asking to do auditory stuff, so I'm sure we'll do it.
It'd be interesting that, like, if you have a congenitally blind person who has to navigate, obviously they learn how to do it, and they're not using visual cues, so there has to be some sort of input from the auditory system, likely.
Yeah, so auditory system for sure. So olfaction, they do use their olfaction. So there are blind mice that still have head direction cells.
3 of them, right?
There's 3 of them. They still have head direction cells, and if you, interestingly, if you lesion their epithelial cells in the olfactory bulb, that kills the head direction cells.
So they're totally navigating by olfaction then?
By stereo olfaction. So there's a new preprint that just came out showing that. So if you block the nostrils, well, the animal can't breathe. But if you lesion both nostrils, you'll lose your head direction tuning. And if you take a tube and you merge them, so now instead of getting 2 signals, it's getting just 1 signal.
Yeah.
That disrupts head direction cells. So it's really even the stereo olfaction,
Yeah.
gradients in smells.
That's cool.
Yeah.
Yes.
Does the age of the mice matter in how they respond, or did you check the longitudinal age, or
For which?
the first
Yeah.
for this one?
So no, we used young, healthy mice for that. I would like to do this in the Alzheimer's animals or some aging studies. Because, we showed that head direction cells are fine, but there's other things that you can look at, like maybe the system doesn't reorient properly. And that could explain why people are getting disoriented in Alzheimer's disease and things like that. So we should do older animals and potentially even Alzheimer's studies.
You had mentioned like an application of this, specifically like studying the amyloid plaque in the mouse models. That like by looking at the medial septum, or not that, looking at the grid cells was because that's like the first sign of degradation. That would be a more accurate way of determining like early onset Alzheimer's.
Early onset, yeah,
people.
exactly.
Okay. How, like, I guess, would you connect it to like the topic that you were talking about before that? I'm sorry if I missed it, but like, would that also be connected to the long-range GABAergic projection?
So,
like,
yeah,
the environment?
you're right to think in this direction because the results are very similar for the Alzheimer's study and for the medial septum study. Grid cells are disrupted, head direction cells and place cells are fine. So it might be that in our Alzheimer's model, there's a degeneration of this projection. I don't know. If that's true, we should be testing it.
Now that you said long range, how do you know it's that these are GABAergic projecting to hippocampus and no
Yeah.
local inhibition that then affect the excitation?
Yeah, so
Viral.
we don't know. We don't know. We're transfecting all of the inhibitory cells, and there's 3 or 4 classes of inhibitory cells in the medial septum. So we've gotten specific, but not the. Maybe we need to go another step further and use like a retrograde AAV.
Right. Any other question? No? Okay, thank you again.